Hi, my name is Rui Zhu. I'm from Indiana University, Bloomington. Today, I'm going to talk about our recent research, privacy-preserving construction of generalized linear mixed model for biomedical computation. We're going to talk about the background about generalized linear mixed model and GWAS first, and then we'll talk about the motivation of this research. And then we will give the big picture about the workflow of GLMM and CGLMM. And then we will give more detail about our main contribution, CGLMM. And then we will talk about our experiment results. The last one, we're gonna talk about the conclusion. So, generalized linear mixed model, or so-called GLMM, has been widely used in GWAS. So what is GLMM? GLMM is an extension of generalized linear model, where we can consider some discrete variable like gender or type of blood as random effect. So in this formula, if we remove the random effect part, this will be a generalized linear model. Mostly, the random effect are those factors that do affect the model, but we don't really want to consider. For example, in GWAS, we are interested in association between some response with SNPs, not gender. But gender do has effect to our prediction sometimes. And then, what does GLMM do in GWAS? In some research, we want to understand the association between some SNPs and case control group, or cancer stage 1 or 2, for example. In here, we give a chart to describe this. We can see the reason why we want to have random effect part in here, for example, male or female, because male or female may have different cancer stage rate. Directly using SNPs as a variable cancer stage 1 or 2 as response to build a generalized linear model will provide an insufficient association. Let's briefly talk about how to optimize regular GLMM first. The whole optimization process is an expectation maximization algorithm, an EM algorithm. Well, the E part is a Metropolis Hastings sampling method, which fixed the fixed effect coefficient beta to optimize random effect Z. In here, the proposed function AZZ prime, it means the probability of Z should be replaced by Z prime, where Z is sample point from the last iteration, and the Z prime is a new sample point. After update the new random effect coefficient, then jump to the M step, which fix the random effect Z to update the fixed effect coefficient beta by using Newton-Raphson algorithm. In here, H is a Hessian matrix, which is a second order derivative. It determines the step length of the update. The first order derivative F prime in here is a vector which determines the direction of the update. This is a typical second order derivative optimization method. So we can use Newton-Raphson to update our fixed effect coefficient beta. So after update our fixed effect beta, we then jump back to ESTEP again. So do this one by one, iteratively until converge. That's a big picture about the regular GLMM optimization. Well, GLMM has been widely used. One typical problem is there are many data holders, or we can call them client or party. And for each data holder, they only has some small amount of data. Using this small amount of data cannot build GLMM model precisely. For example, in here, we have three party 1, party 2, party 3, and they hold their own private data. So also in GWAS, data are highly sensitive. Data holders cannot share those data directly. More specifically, this scenario is so-called horizontal partition, which each party holds some private data. Those private data has same feature, or for example, SNPs. They are a subset of the subject, the entire data X, but different observation or record. Or in here, we can see each party holder holds several different lines of entire data X. You can see party 1 holds several lines from the entire data set X. So note that in here, X1, X2, and X3, which from party 1 to party 3, can build CGLMM individually. So our goal is to develop the same CGLMM which using entire data X, but without share each individual data. In regular CGLMM, those private data in the right-hand side need to merge to a large matrix so that we can solve the CGLMM like in the previous slide. However, the challenge is like what we just talked about. Each party cannot share their own data. So that is impossible to merge X1 to X3 into a big giant matrix. In our research, we propose a new collaborative algorithm in order to solve this problem, which each client do not need to share data directly. Instead, they only need to share a homomorphic encrypted intermediary result so that data holders' privacy then can be protected. We call this new collaborative method as collaborative generalized linear mixed model, or just CGLMM. The big picture of CGLMM is like this. So right-hand side is a CGLMM compared with the left-hand side, the GLMM. Similar with regular optimization of GLMM, in the left-hand side, it used an EM algorithm, where ESTEP is a metropolis hasting algorithm to update the random effect. MSTEP is a Newton-Raphson used to update the fixed effect. The CGLMM uses the same workflow, but the progress of of metropolis hasting and Newton-Raphson has been changed. For example, here we have 3 parties in the system. In both E and M step, each party sends a homomorphic encrypted intermediary result to central server. Server use those results to compute the update, then send the update back to each party. Then start the next round. So next, we will talk about more detail about E and M step in CGLMM. The E step is a collaborative metropolis hasting sampling. The goal is fix the fixed effect beta to update the random effect Z. In the beginning, central server on the right-hand side send current beta to each client. The beta comes from previous M step, or it could be the initial beta if it is first round of EM algorithm. Then each client uses beta and their own data to do a private computation to get an intermediary result, AK. Send this result with homomorphic encryption to central server in order to get a global result. The only thing that server needs to do is a joint computation, which is just a matrix summation of those results. And so server send this result with new sampling point back to each party to start a new iteration. Note that Since the only thing that server need to do is the joint computation of homomorphic encrypted data, then we can use parallel homomorphic addition to deal with it, which is much more faster than fully homomorphic encryption. The ISMB is a collaborative Newton-Raphson algorithm. The goal is to fix the random effect Z and update the fixed effect beta. Similar with metropolitan algorithm client is a homomorphic encrypted intermediary result Hessian matrix. So this Hessian matrix was computed by using the party case private data at first order derivative. So note that those are all intermediary results. So send those intermediary results to central server. After collected all results, server do a joint computation. One matrix summation for Hessian matrix and one vector summation for first order derivative. Then the fixed effect will be updated. Send the new beta back to each client to start the new iteration. Also, note that no matter ESTEP or ISMB, central server only need to do a joint computation, matrix summation or vector summation, which means we can also use parallel homomorphic addition. We implement CGLMM in R language, and the source code has been published to GitHub. There are several important dependence packages we used. For example, in here, in order to send homomorphic encrypted data to server, we used RSOX for simulated data transmission. And in order to implement the parallel homomorphic addition, we use homomorphic R. And here's a link of the our source code. We did the experiment in two parts, simulated data and real GWAS data. For the simulated GWAS data, the data set has one random effect with 5 different levels. And we simulated 3 data sets for different amount of SNPs, 50, 100, and 150. Response Y is 0 or 1, so which is binary case control. X is simulated SNPs. They are either 00, 01, or 11, which means homozygous major, heterozygous, and homozygous minor, respectively. The generated probability of those 3 types are 49%, 42%, and 9%. For the real GWAS data, we used are from the 1000 Genomic Project. Similar setup with simulated data. We have 3 data sets, respectively, with 50, 100, and 150 SNPs. In each data set, there are 10 SNPs are significant. And all the data set has 2000 record. The response Y is 50% positive and 50% negative. Also, in the real data set, the random effect only has 2 levels, male or female. And here's the ratio about the female and male. So almost 50-50. So we want to compare the performance difference between regular GLMM and CGLMM to see whether the performance similar or not. In here, we show the graph of the performance when applying to simulated data set 2, which has 100 SNPs. Because other two data set performance pretty similar. We didn't show all the picture in here. The first picture is the update magnitude. The x-axis is a iteration. We have 5 iterations in this experiment. And we can see the GLMM and CGLMM performance pretty close. And second graph is the random effect coefficient value, which is sigma. We can see it's also pretty similar. And the blue line is the true sigma. Since this is a simulated data, we know the true sigma. And the last graph is we have 100 SNPs, so we have 100 beta. So we can see the GLMM and CGLM performs pretty similar and pretty close. Also, the true beta are pretty close to them. As well as the real data, the graph we present here is when we're applying real GWAS data set 2, which also contain 100 SNPs. And no matter the update magnitude or sigma or beta, they all performance pretty similar, pretty close. So we proposed a collaborative method, CGLMM, to solve GLMM with horizontal partition. The method is mathematical equivalent with the regular GLMM optimization. Also, we implement CGLMM in R language, and the code has been published to GitHub. In the future, we may consider about vertical partition, which each data holder has same observation or record, but each party has different SNPs. The research was partially supported by National Institute of Health grant and Indiana University PHI. We thank Yuebu for her help of using the human genomic data from a 1,000 genomic project. Thank you.